Hello, Susan and Tharun. Let's wait for a few more people to come show up.
Can you hear me now?
Yeah, I can hear you.
Okay, I had to get the right microphone. My camera's not working today.
Oh, yeah. Sorry about that, but yeah, seems like your camera works when your microphone does, and vice versa. And
yes, this is a gamer computer, and it doesn't have a camera on board.
Oh, okay.
I have to plug one into USB port, and then other things don't work.
Huh. I don't know.
Yeah, don't know why.
Hello, Shruti.
Hi everyone.
Hi.
Hello.
Hi Susan, how are you?
I'm good, except my camera's not working today.
Oh, because your mic is working, that's why maybe?
Yes, that's right. One or the other. Yes, I know. I need them both working, but, well, I'll have to work on it. I don't even know where the camera went this morning, so, yeah. Oh, there it is. I found it.
Oh, okay.
If I plug it in, though, my voice will disappear.
Oh, okay. Well, then probably be better off just to have your voice, because you say something.
Yeah, I think so.
Yeah.
Just for today, yeah, because otherwise I use a Bluetooth microphone to communicate.
Yeah. Okay. Well, I guess I don't know who else is going to show up. So welcome to the meeting. We have a couple of things to talk about before we start. Tharun is going to give a demo, or he's going to show us some results he got from the Bacillarea work. He just put it in Slack recently.
Even I have something, I was just working on something, so if it is possible, I would also like to present.
Okay, yeah.
It's regarding speech motion detection.
Okay, that sounds good. So yeah, Tharun will talk about the Bacillarea stuff, and he's been in contact with Ujwal about it. So Ujwal is actually going to be making the meetings soon, but he's got another conflict. So he told me about that, and so he's still going at it. He's working with Tharun on this, and then Shruti will present on her topic, and then I don't know if Krishna is going to show up because we have some things maybe to talk about some submissions, but we'll see. So a couple things before we. Oh, go ahead.
Oh, okay, sorry. Thank you for telling me about the gastrulation talks.
Oh, yeah.
The one about the ants is perfect. I might be able to use it somewhere, somehow.
Yeah.
With my thesis or one of my projects.
Yeah, that was good. So, yeah, last week I presented on ants and plants, and then last week, later in the week, there was a gastrulation seminar series that's online, and they talked about different types of embryos, ants, and things like that. And that's why I kind of did the ants and plants, because I had seen that coming up, and I thought, well, that'd be a good topic to get into. So Susan saw the gastrulation seminar on that, so that's good. It's not something that we usually hear about, so that's why I thought it would be a great topic. So,
It was a different model organism, one that they had used, they said, since the 1950s. It was used prior to that, so they thought, why not? And it's given them some interesting results because the shell of the egg is used in gastrulation.
Okay. That sounds good. And then, we might actually talk a little bit about that, like following up on that. I don't know if they're like, we might go down a sort of a rabbit hole on that. If we have, if we can think about some things we might want to know more in depth about it. I'm not really familiar with the model organisms there, but we could look into it and see what they have. So that's good. So I guess why don't I start off with a couple things here. Let's see. Let me share my screen. Actually, we have two things in the chat. Okay. So Tharun's not prepared to do it this week. But we can do it. We can do it next week. So let's see. Let me do a screen share. All right. So the first thing I'll point out is congratulations to Mayuk and the people working on the DevoLearn release. So this is I think Mainak is also involved in this. They did this stuff that Mainak was showing on the training data, so they were able to expand the resolution of the images and add additional training data. And so Mayuk combined this into a release of DevoLearn. So this is 0.3.0. So last release was, I think, 0.2.2, and then now he's going to 0.3.0. Again, I told him the rules for this are that, every time we get some sort of major addition, something that isn't like a very small commit, to release a new version. And so he's got this release. This was about 7 hours ago, so it's pretty recent. And this is the PyP page. So this is PyP, where you can download open source platforms written in Python. And so this is we still have it on GitHub. It's just that this is it's like a public repository for Python applications. And so you can see everything is pretty much the same, except it's improved with those changes. And so this is congratulations to everyone involved in that. Mayuk also contacted me and asked me about different types of... He's interested in sort of working more on DevoLearn in terms of the C. elegans literature. So I pointed him to some papers here, and I'm starting to create a bibliography on this. So if people are interested in contributing to this bibliography, let me know. This is called Spatial Distributions for Deep Learning in C. elegans. And so the idea here is to have a repository of different papers on C. elegans, on like analyzing spatial distributions of cells in embryogenesis, with sort of a focus on deep learning applications. So we have a folder in deep learning papers. So there are a couple of papers here on what they call 4D analysis. So this is, 3 dimensions of space plus time. And we've talked about 4D analysis in this group as well. So this is it has a lot to do with the cell tracking, which we've talked about, and the 4-dimensional analysis, which are the 3 dimensions of space plus time. So you have all these images where there's a process unfolding. And that's one folder. And then we have biology and computational biology, which is this. We have a bunch of papers here that are more biological in nature. So things about different aspects of embryogenesis, embryo networks, asymmetric cell division, gastrulation. There's a transcription lineage atlas, so we have some molecular information in here as well. And so that's so if we want to keep growing this bibliography, it's open. I can make the link open. Just let me know if you want to contribute, and I can put them in or give you the link. And so it's pretty early days, but Mayuk said that he found it to be pretty useful. So there's the repository. Hello, Krishna.
Hi, Bradley.
How are you?
I'm good. How are you?
All right. So I was just going over some of the news from this last week. Yeah, I think that's it for now. Well, actually, I did want to go over the paper submissions. So we've been talking about paper submissions, and I want to go over this really quickly, just to give us an idea of like what's due soon and what's not due soon. So the Evolution Conference are the first two entries on this list, and I found out more about this abstract submissions for Evolution, and apparently they don't open until March 1st. So I guess I gave the impression last time maybe, or I was under the impression that the deadline was March 1st. But it's actually the submissions that open on March 1st. So I don't exactly know what the deadline is, but that means that we have more time, so that's good news. That means we can, that's when we could start submitting things, so we have more time on that. We have a couple more weeks probably. We have this Kill the Winners. That's something that that Krishna is interested in, and I'm on that as well. And if you want to be on that, we can, incorporate people's contributions into that. We also have the Euler Paths for Life. That's the talk that I showed I'll maybe talk about the abstract later on. That's also something for that conference. And if you're interested in submitting something to Evolution 2021, you can add it to the list or tell me and I can add it to the list. The Flash Talk, we did that already. The DevoLearn paper, that's in progress. And we have this, the library I just showed you might play into that, making that a more substantial paper. But we'll get into that. That has no deadline. I mean, we might submit it for something, but as of right now, it doesn't have a deadline. So there's also this growth form and theory of deep learning abstract, which we've been working on, and that's kind of stalled out. But if we find a venue, we'll submit it there. We have the Google Summer of Code stuff, which I know people are working on. And so those projects are still, I don't think the application period is opened yet for that. I need to go over a couple people have already sent me proposals for that to review, so I'll review that. I think Mainak sent me one, for example, so I'll review that and look it over. And then I know we talked about having a separate session for onboarding for this. So the deadline is a ways off on the application period. So I think once the application period actually starts, which I think is at the end of this month, at the beginning of next month, then we'll start to maybe have those onboarding meetings. I don't know if we need to send around or we need to kind of settle on a date and time for that. So I think Krishna was organizing that, so I don't know if he has an update on that or what his thoughts are.
Yeah. So can I start presenting?
Oh yeah, let me stop presenting my screen here. Okay. Go ahead.
Hi Susan. Hi Shruti. Hi Tharun. Hi Mainak. So 1 minute. Did you see that, Brady? Did you see that evolution paper that I sent you?
Yeah, I did. I couldn't. I mean, I don't have it up right now, so maybe you can bring it up.
Yeah. Oh, is my screen visible?
Not yet.
Is it up?
Not yet.
Okay.
There we go. Here it is.
So here we go. Okay, so first of all, regarding that onboarding meeting, that is, these are the slides that I have made.
Yeah.
Sorry. Oops, 1 minute. Okay. So, if anyone who is interested in GSOC, you can tell them, or you can put on your Slack, your LinkedIn, that what are we and what we are doing, and it's what is open source. I guess if a person is applying for GSOC, he must be knowing few things what open source is, and one how one can contribute in it. And as far as DevoWorm is concerned, every contribution is welcome, even though if no matter how you can say how small or big it is, every contribution matters and it is welcome. And why a person should contribute to open source, like it also promotes the fraternity, the you can say the community thing, and also one gets a lot of things to learn from. Like when I joined this group, I learned a lot of things from Bradley, like, and from Susan and from Dick. So yeah, if a person joins an open source community, he learns a lot of things. Just a meme. And what is Google Summer of Code? It's a program it's a 10-week programming project. Earlier it was it was of 20 weeks, but now Google has shortened the duration. And here it is the timeline. And here it is about DevoWorm, what we do, and how we are working on computational biology, artificial intelligence, and data science. Our main, you can say, core thing is this, but we are not limited to only computational biology or developmental biology or AI. We are open to mostly everything, if not everything. So, and how a person can contribute, you just have to look for the thing that you want to work on. You have to see the guidelines and how the thing was made. Then you have to find a point where you can create a patch, and you can improve, enhance the thing, or you can reduce the bugs, and you can fill an issue and then submit the change. So this is the onboarding presentation.
Yeah. So this.
So was it okay?
Oh yeah, it looks good.
Yeah. Okay. Now second thing is the evolution paper, and I'm sorry that you are not able to open that. I send you in the doc format only.
Oh well, I got the doc attachment, but I don't have it open here. But the other one, I don't know if this is the same one. You sent me a paper.
I think I sent you regarding the different one. Okay.
Okay.
So here it is. It's the paper regarding the evolution conference. I hope that we only need to submit the abstract. I guess a 2 pages abstract is fine. What do you think, Bradley?
Well, I don't know what the guidelines are. So if it's smaller, then we can just condense it down. But I would keep this and expand this into a paper, because we can submit it to different venues. So if you write a paper and you have an abstract, you condense it down to an abstract, then that's your abstract for the paper, and then you can submit the paper somewhere else later. So
Yes,
it works out. Yeah.
Okay, now what else is left? Okay, so I send you this paper, that extended abstract for differences between biological and artificial neural networks. It's the extended version of our NeuroMatch conference.
Right.
Yes, I think Jesse was talking about something on the that orthogonal meeting.
Right.
And I don't know where we are going to send it. I think for neuro What was the conference name?
Well, there's Artificial Life. That's the one that's coming up, so we can submit it there. Because we're already submitting other things to that. So that might be a good one. And that's 2 pages of their format. So this format is 1 column. Their format is 2 columns. Okay. And so it's a little bit, it gives you a little bit more space. But we'll figure that out. Just try to get it written up in about maybe, say, 3 of these pages. And then we'll fit it into the template.
So, a single page divided into 2 portions.
Well, yeah, we have to kind of, the format is a little weird, so it's not. I would just start with this, and then maybe do 3 full pages of this, or maybe a little bit more, and then we can get it into a 2-page format, which is because the font is a different size, and so yeah.
I tried to follow the regulation that I guess. Once my paper got rejected because I was using different font.
Yeah,
it's this Comic Sans, and I like to make it funky, and they rejected it.
Yeah, yep.
So I'll just have that New Times Roman maybe. So the third thing is GSOC is done, evolution is done, difference between A and B. Okay, so is Jesse here?
I don't think so. Okay. Oh, so this is from our Saturday meeting where we have some, So this is the idea that you had for that.
Okay, yes, it is. So, I'll try to, make it as short as possible.
Okay.
So, the problem nowadays, that there are a lot of complications coming with artificial intelligence. And most of them are related to the bias and the ethical issues. So as we know that AI safety is very real, and most of us are not following it. Not us doesn't mean that we regular researchers, but big companies. I think that you all might have heard about an AI researcher was fired from Google because she was whistling blowing some of the bias, underlying bias. And even myself, I have, I'll not be naming that company, but one of my senior told me that he works in, one of the fan companies and how they are manipulating the AI data. So here it is garbage in and garbage out. It's a popular term. So how we put, if our input is faulty, then our output will also be faulty. So what is the feedback loop? Whenever a machine learning model is being trained, including the feedback loop is helpful, as it lets the algorithm make minute corrections. So it is a process how the algorithm really learns and how it corrects itself. So what is the problem with that approach? The same garbage in, garbage out problem. If it's learning from itself only and it is committing some mistake, then once it tries to learn from itself, it will be learning from its mistakes. So, random meme. I like to put this, lighten the. I usually get nervous while presenting something, so that's why I used to like to add them. So here it is an example of positive feedback loop. So for example, we have a person and he doesn't have a good credit score. And then we say that he has high chances of being a defaulter, so he gets high interest rates on his particular. So and since he is getting higher rate interest, there are more chances that he will not be able to pay his debts. So that that there is a, you can see that we are initially assuming that he won't be able to pay it, and we imply some condition, and then easily he won't be able to pay it because he is getting higher rate of interest. So at the first time, if you have induced that bias and kept the rate of interest normal like other people, there are chances that you might have been able to pay the debt. So if you is already starting 2 steps back from a normal person, there are chances that he won't be able to pay it. And here is one more example. Like if we have historical records of this neighborhood, and we see that this neighborhood has high crime rate. what we will do, we will employ more, you can say, police persons. And since if there are more person, they'll know that okay, this is an high risk area, so they'll be more conscious and they'll be overcorrecting it, like making more arrests. And since now more interests are made, again that we have that statistically we classify that area to be now critically risky. And now we have more police personnel deployed. They will be they will be more cautious and they will try to overcompensate it. And what will happen now? Again, more interest would be, arrest would be made. And I read an article on COVID-19, and it was found that 80% of the people of a certain hospital, 80% of the patients who had asthma, they were not, they were not, they didn't become critically ill. Whereas the people who were normal, who were healthy, they had more chances of getting critically ill in the treatment. So how it is possible that people who were already sick were becoming healthy, and healthy people were having you can say larger probability of getting sick? Because people having asthma were very cautious, and doctors treating them were over cautious. So if even they have slightest of medication complication, or they were not responding to the medication, or even if there was slightly, you can say, difference in their vitals, doctor were very, you can say, cautious. Whereas in the case of normal person, they were little bit lenient. So here, they say that if we already, have something in our mind, if we make up our mind for something like that, the world seems to be in that light. Like if, I guess it reminds me of your, psychological discussion that you were telling me, Bradley, that how phobias were overcome. So, like if we make a perception of something, the world appears to us in that way. So if humans are inducing this stereotype and this bias, machine learning should not be, inducing it from us. This is it. Then, so a possible solution to that problem is to, involving some human beings. And I have one more example. Like for example, if arrests are made and we have 26 letters, and we somewhat find that people who were arrested, majority of them are having their names start with letter A. So we will, our machine learning algorithm will learn, okay, people whose names start with A are more prone to be criminal. But that is not true. That is just a random pattern that is generated. So when we are using AI, we are trying to find patterns. These patterns should be, have some meaning. There can be a lot of, you can say, random patterns generating, like if a person's name starts with A, so we can say that only A people are criminals. That's not true. That's just a random pattern that emerged. So a possible solution to this problem is involving human beings, and we can correct the misjudgment of the machine. So that's it. These are the references, and I'll suggest that Anna Coop's lecture on positive feedback loops and negative feedback loop is very interesting. And if if you people are interested in these things, kindly have a look in this. That's it from my side.
Well, that's very good. I'm glad that you were able to get that together. We were talking about at our Saturday meeting. We were talking about this. There's a project that Jesse, who attends this group, is doing on AI ethics and that sort of thing. So we have an interest group over there if you're interested in joining in. But we also should probably have a component of this. The DevoWorm ML materials that we have. So we could add in a module on this as well. I mean, I'd like to make it more like, kind of general, like, data, how do you, if you find a pattern in your data, like for the AI ethics group in the other meeting, this is great. For the DevoWorm ML, I'd like to focus on, like, if you find patterns in your data, how do you overcome them? How do you identify them if they're spurious or if they're... So, I mean, this would be good. This would be a good addition.
Daddy, I have one more example relating to
Go
this.
ahead.
I mean, this is an amazing topic. So I read somewhere that due to data in America which we have for criminals, almost 7 to 8 times there are black people who are reported as criminals as compared to white people. So the model will learn to recognize black people as it will have a bias for black people to be criminals even if they are not. And of course we also know that black people have a little, we human beings also have a little more bias regarding them, that if there is a black person and a white person, a black person might get a little more punishment as compared to black. So that was discovered, and it was said that because of this positive feedback loop, what was happening was the model was predicting black people to be criminal even for a very inconsiderate amount of crime which they did. And because of that, in that particular area, there were more black people obviously in jails, and this led to a kind of chaos, and it was later detected, it was because of the positive feedback loop that Krishna was talking about, where the bias caused more arrest of black people as compared to white people, because the data that they had already contained such values. So it can be...
it's the same marijuana thing that I was telling you on
Yeah.
Saturday.
Okay.
Yes, I've read something about that.
Yeah.
I read a paper about that.
Yeah, this was something that happened, and it was a little shocking to people. And
Yeah, sometimes, people are not really racist, to be very honest, but these are, some of the perceptions that are get from wrong input that leads us to a...
And maybe historically it might be true, but we can't take that further, right?
That historical thing.
Yeah.
Mostly, this bias is of, you can say that even a slight depreciation in our input value can largely affect how our output looks.
Yeah, it can be exponential.
Get it? Something like that.
This brings up another topic too that we haven't talked about, and either group actually, is the idea of having like these human AI hybrid systems. So like, to be able to put humans into the loop somewhere and to say, is this a good judgment that the algorithm is making, or is it like something that I have a talk on this. I haven't presented it in this group or in the other group, but I might present it at some point. So if you're familiar with what they call hybrid chess, or like the what do they call them, the man and horse combinations, I can't remember what they call them. But do you know what I mean? Like
Yeah.
Okay, so there's this type of chess where they play. So
I'm remembering Narnia.
Yeah. So like in the 90s, Deep Blue was able to beat a human. It was able to beat Garry Kasparov at chess, and he got upset about it. So he went off and he figured out a way to like incorporate like machine learning. It wasn't called machine learning at the time, but to incorporate that into playing chess. So like the human would supply some of the innovation and technique, and the machine would then execute the moves. And then that was like a different type of chess that they invented in the aftermath of a machine beating a human. And of course, the human AI hybrids can do quite well and sometimes beat machines. I don't really know what I don't really follow the field, but like it's actually an interesting question. And of course, you can use this kind of hybrid systems for other things as well, and there are a lot of advantages to it, because you have things that obviously humans can say are obviously right or wrong, and machines can't do that because they don't have a lot of the context that we do and things like that. So it's definitely an interesting topical area. So thank you for presenting on that. I think we'll go. Let me finish our list, and then I'll go to Shruti, and then we'll do her presentation.
Yeah,
Okay.
sure.
So thank you for that, Krishna. We have Mainak said, nice presentation, Krishna. So let me share my screen again. So we're going back to the paper submissions. So that's Krishna's stuff. So he's got the evolution abstract. He's got some other things. I don't know if I put the A-Life submission on this. I have another document from my other group, and I may have put it on there. But let's just pencil in this abstract that Krishna showed, the 2-page abstract on the BNNs and ANNs. Yeah, I won't put it on this list. I have it on the other list, though. So that'll be submitted to A-Life conference, and the tentative deadline for that is March 7. So that's coming up, but you know, it's not that much. I think you've got a lot of it in there. We just need to revise it a bit. And maybe next week we'll go over it a little bit more detail, and kind of walk through it a bit more. So then we have this. Then we have some papers that we want to submit. So there's this bacillary non-neuronal cognition paper. That's something that has been accepted as a book chapter in the proposal form, and then it's due April 30th. And I think we're far along on that, but we are going to turn our attention to that in the coming weeks, and you know, kind of fill in the gaps, because I think we have a good I think we have a good basis for everything. Like everything's in place for it. It just needs to be fleshed out into a actual chapter. The Elegans conference is coming up on the deadline for that is coming up on March 25th. Excuse me. And this is the submission portal. It will let you know that they charge a fee for submitting an abstract, and there's a fee for attending the conference, but it's totally online. So if you're interested in attending a C. elegans conference, you know, you can sign up or submit something. I'm not planning to submit anything in particular for this, but we can talk about that. The Euler paths for life. This is the abstract that I want to submit to evolution, and that's more of a biological abstract. And then there's this full paper which is to be submitted to CompleNet, which is a network science conference, and that's the 26th of March. So these are the links to the CompleNet conference, complenet.weebly.com. And I actually have a couple of, I think I have the abstracts in here. Okay. So the way I'm doing this for this paper is Euler cycles for life. And I might kind of go over this really quickly later, like the slides, to remind you what it's about. But I have two versions of this. One is an evolution of development approach, which is more like biological language and a more biological motivation. And then the network science version, which is more network science-like. So if we go into the evolution of development version, we see that there's it starts the evolution or transmission of multicellular life in the form of cellular aggregates and embryos is marked by a need to inform internal networks for oxygen and nutrient transfers between cells. So there's a focus on the functional aspects of these networks, and then tracing the networks with these Euler cycles. And again, I'll talk about I'll kind of briefly go over what that is later on, but we're going to use Euler paths, which are a tool from graph theory, to evaluate these networks that exist just between the edges of cells. So, you've seen the embryos, for example. They have cells, and the cells form junctions between each other. Well, they form boundaries between each other. And along those boundaries, there are all sorts of gap junctions and other passageways between cells. And then we're evaluating those edges to see if you can form a continuous network, which implies that there's some sort of equilibrium network for transferring things from the edges of the multicellular aggregate into the middle of the aggregate. So that's basically the motivation here. And then this kind of goes into how you can sort of simulate by doing this exercise, simulate the establishment of new phenotypic modules. So there's an emphasis on modularity. But then the network version, which is a full paper, focuses a little bit differently. And that is, again, we're trying to form these networks, motivated a little bit with biology, but then we kind of get into this we kind of use some graph theory language here to describe the graphs, and this kind of describes the mechanism, and this actually the thing we're going to focus on in this abstract is where you have these meta-replication events, where you have a unipartite network that becomes a bipartite network, which is different than sort of the assumption of a module, phenotypic module, but it's very similar. But I'm going to kind of use different types of language with these different submissions. So what I'm trying to show you here is that when you submit something to different venues, sometimes you have to change the language a bit to get people, to get people's attention to make it relevant. But that's going to proceed. Simply just submitting an abstract, of course, means that you're going to create some slides later. So the slides are going to focus on the biology, in this case, for the evolution conference, but on network science for the ComplNet conference, and graph theory too. So that's my lesson for the day on that. So I also have this other idea, and I've submitted this to Networks 2021, which is it's also a virtual conference. It's network theory. It's I don't know if I don't have a link to the abs. Oh, maybe I do. Yeah, this one here. So this is an abstract that I submitted to this Networks conference. I'll probably submit it to ComplNet. This is about embryo networks as generative divergent integration. So we have these things called embryo networks, which we published on a couple of years ago. And I presented on this to NetSci a couple of years ago as well, to a conference. And the idea is that you have that embryos form these networks. If you look at the different cells in an embryo, they interact with one another and they form these networks that you can define through graph theory using thresholds, and they form these complex networks. And the idea is that they form networks and they form structure over time as cells divide and move around in the embryo. And they form these modules. As I mentioned before, they form phenotypic modules. They form like highly densely connected cells, meaning that they're cells that are very tightly packed together. And so all these things happen. But in, say, like something like C. elegans, you have these developmental cells doing this. And then you also have a nervous system that's starting to form. So the nervous system, of course, forms this connectome, which we all know from the C. elegans connectome literature, that you have, like, basically as the neurons begin to differentiate and form, functional neurons, they form these neural networks that have connections between them. And so what I'm talking about in this abstract is the formation of these two networks from a single network. So early on in embryogenesis, you have this one network that's basically the proximity of different developmental cells, and maybe some gap junctions in between them that form connections because they're neighbors. But also start to get like sets of neural cells that are going to later function together into a neural network, into a connectome. And so you get this divergence into two different networks. And so this abstract talks about this and this process and highlighting this process. So this is going to be a talk maybe at NetSci and probably also an abstract at ComplNet. So that's something we can talk about later. I just wrote that abstract, I think, this week. So this is there's more to come on that. More immediate, though, we have the biosystem special issue that's on March 1st. So we have a couple papers for that. We have the temporal paper, the bursts paper, where we're talking, the one we've been talking about with C. elegans and zebrafish. And there's some trivial revisions to that. So I'll probably go through that. And again, if you want to contribute to this, one way you can is by finding a good open access or fair use animation of of embryogenesis, especially like a time series sort of thing. So that's what I'm trying to find now.
I'm contributing to the biosystems papers as well with my 3D ball microscope. Probably nothing more than, it takes 3 images from 3 dimensions and compares how this compares with previous attempts.
Yeah.
Including my flipping microscope. So I still don't have the 3D software to project all of this onto a sphere. So just saying, I am contributing, but it's just here's a microscope. It needs to be automated, and it needs to be and the software to project the images needs to be developed. So it's just like this is the first step.
Right.
Anyway.
Yeah. Well, that sounds good. Yeah, and I know that Dick has a paper on, or we were talking about the paper on the boring billion, which is also part of this special issue. So I didn't put that on here, but that's also another thing. I'll just put that there instead. Okay. So I'll put that. Yeah, I'll put this. So anyone contributing anything to that, the deadline is March 1st, so it's coming up. I know that Ojwal and Jesse are currently co-authors on it, so I wanted them to get a heads up on it. We'll talk about it more this week. Then there's this Amazon book, which we talked about last week. That was something that Krishna was spearheading, and we'll talk more about that in the coming weeks. And then finally, there's this INCF Neuroinformatics Assembly coming up. And this is the INCF. These are sponsors for Google Summer of Code. But they also have this assembly coming up, and we need a 1,500 character abstract for that. So I think one of the things we might focus on for that is maybe some sort of, some sort of DevoWorm or DevoZoo epistemological directories mashup. And if you don't know what epistemological directories are, it's a thing that we're working on in my other group. I think if we do that, I'll kind of maybe give a presentation about that in this group a little bit to give you an idea of what this is, and then, write up an abstract. So if you're interested but this isn't for sure. I don't know what we're going to do for this, but if you want to participate in this, let me know. I think this will be a good opportunity for INCF to see what we're doing. I know I presented last year at the GSOC Mentors workshop, and so they were pretty excited about that. So that's a good opportunity as well. So let me unshare my screen now, and let me look at the chat. Okay, yeah. Krishna says, I add jokes to catch attention. Those are your memes that you put in your talks. They're very nice. So Shruti, do you want to now present on your topic?
Yeah, I will.
Okay.
Yeah, so is my screen visible now?
Yeah.
Alright. Recently I have been working on a project which is called speech emotion detection. It is basically taking the speech, passing it through a deep neural network and detecting emotions. But the thing here is that what we are considering here is that the fact that voice often reflects the underlying emotions through the tone and pitch to which we speak. Like if you are happy, we'll have a cheerful voice. If you are sad, we'll have low voice and stuff like that. So the data set which I took was of Radhi's data set, and the emotions you can see here. First of all, the emotion is a very, very complicated thing. We as humans are sometimes unable to find whether the person is actually happy or sad. And psychology also finds that this one few of the most difficult concepts. Emotions, there are two kinds of emotions: categorical, which can be discrete, and dimensional, where are continuous stress and strain, strain and relaxation, arousal and substrain, pleasure and unpleasure, and they are continuing in a sense, and the other one are categorical, which are quite discrete in their approach, like anger, disgust, fear, happiness, sadness. All these can be, quite properly distinguished. So the question is, why do we need speech emotion detection? So speech emotion recognition is something which is worked on using machine learning, but deep learning techniques are not used that much till now, but they can definitely provide better results. It can be basically used in the medical field. For example, if we can say, like right now, especially in the COVID times, where people don't prefer going to hospitals for small petty reasons. So if a person is actually very sick and hyper and he's anxious, that thing, if the machine beforehand could detect, okay, the person is a little bit anxious or he is, not in a very good state of mind. So if there is a possibility, we can put it on a more urgent base and can try to give it a priority over others. Like same can be considered in the customer care services and the recommendation system as well. Another example would it not be great if Alexa would understand that, okay, this time we are irritated to talk something like this and not just keep on babbling and babbling. If Alexa would know, okay, now the person is happy, so she'll be in a cheerful mood and go with the flow kind of thing. So I guess this emotion detection from speech would be helpful in Alexa or Siri and all these things too. So the phase of my project was first of all the data set selection. Then I had to extract features, like how would my deep learning model learn about the emotion underlying in the speech. Data augmentation, because the data set that I have is a little small, so data augmentation was required, and finally classification. Yeah, so like I was saying, the data set that I used was called the Ravi's data set, where we have 12 actors and actresses. So writing kind of audio we get, and we have 8 emotions like you can see. Most of them are fairly distributed. The neutral one is like 50% less than the others, but yeah, it's a fair distribution here. So we have anger, we have neutral, we have disgust, sad, happy, surprise, calm, and fear, and almost around 200-ish are the count of them. So yeah, just let me know if you can hear the voice when I'm the audio when I'm playing it. So was it audible?
No.
Okay, I'll remove my. Yeah. Was that audible?
A little bit, yeah. It was very low, but
Okay, maybe. So yeah, okay, the data set contains, maybe the next 3 audios are a little more louder, I believe. So yeah, the data, this audio is saying that kids are talking by the door. And what I've done for augmentation is, because the data set is not that big, so I've added some noise in the data.
Kids are talking by the door?
Yeah, so this was a little bit, if you could, I'm not very sure if you could hear it or not, but there was some noise added to it. Some time stretching was done.
Kids are talking by the door?
The time was stretched, and it was, the length, you can see it was 4 and now it's 5. And the pitch was also scaling.
Kids are talking by the door?
This was basically done because the data which we had was not enough. 1500 data samples is not very huge number for for data deep learning. So yeah, this augmentation was done, and I extracted features. One was MFCC, which is frequency versus time, and the other is a spectrogram, which again is frequency versus time, but its representation is different. Spectrogram gives us the intensity in the form of color shades, and MFCC is in the form of the length. We can say the pitch was, the higher the pitch, the more the length, you could say, from the mean position. So yeah, I've just put in some audio, some clips that this was for one of the sound, and you can see this is the spectrogram which we got, and this is the MFCC which we got for a sad sound. It was a little, you have spaces in between. The darker shades show higher intensity of voice, the lighter one shows lower intensity. This is for the sad one. You could see for surprised one. For sad, you see some gaps in between, because people tend to be a little more slow and stammer when they're sad, and when they're excited, there's a continuous thing going on when they speak. So this were the small details which we might not be able to see so very nicely, but the deep learning model did it. And this is some audios that I tried on the model which I trained.
I'm happy.
Okay, so yeah, this was like audio was like this, and the model predicted it as to be happy, and another was
The sun rises in the east.
this is a little. I mean, you can say it shows disgust, so this voice might be disgusting to the model. So yeah, this is what I found, that using images and not the speech, and plotting the frequency in on the time axis would help the model, enable the model to recognize the emotions. And I've used ResNet 50 for training my model. I've first of all converted my audio into images like I showed you before, and then those were trained in the model, and I got these results from that. So we can say a general thing was MFCCs provided better result as compared to the spectrogram, and CNN are better than other machine learning algorithms for this task because we have a large amount of data, and of course we know convolution neural networks are good at detecting what's in the image. So yeah, so future work would be, we humans are very clever, right? So we may say something which is very good in a very bad tone, or just sarcasm, is, for example. So detecting those emotions would be hard just on the basis of the pitch or how it is looking. So if we are able to detect the words and the speech and put them together, then our model will be like twice or thrice, maybe 5 times better than what it is working right now. So my future work here would be to consider NLP2 and adding NLP in my model, so that it's not just the pitch but the context also the model understands, and the predictions are much more better. So yeah, thank you. This is what I do.
Well, thank you. That was very nice. Mainak had a question, I believe.
Oh, I did not get a question.
No, it actually wasn't really a question. The thing was that like she was sharing her screen and it went away for me all of a sudden.
Oh,
So I just wanted to let her know that. So,
okay.
So was no one able to see my screen?
yeah, we were able to see your screen. It came back. It came back after a short while, after like 10-12 seconds. So maybe it was an issue with my laptop or my network. Yeah.
Any
And
other?
by the way, the presentation was nice. Like,
Thank you so much. Thank you.
it's really interesting. Plus, maybe like you're speaking of further work. Maybe you can have like a live script that will run with, like it will capture the voice or say the audio from the laptop mic and it will classify that. Yeah, absolutely.
The real
And I
time.
guess if you're using ResNet 50, then it's completely possible because it's really quick. Because it's
Actually,
mostly just conv layers.
I've already done that thing. It is done, but it was not having a proper API. I was not having a proper API to present it right now. So maybe in the next week or two, that would work, because real time is exactly what we are looking for. So thank you for that.
Exactly.
Yeah.
Absolutely. And good work.
Thank you so much. Thank you for the appreciation.
Any other questions?
Thank you for the talk. It was interesting. I think that's a very interesting line of research.
Thank you.
And I have noticed when raising children that when things got quiet, that's when you had to worry about things. Yes.
That is what it is.
Jesse said that great talk. We'll have to go back and see the whole thing. Yeah. So Jesse.
Oh, I guess there was something wrong with my connectivity, because I guess no one was able to see my screen for some time.
Well, maybe you could put your slides in the Slack.
I will.
Yeah.
I definitely
Okay.
will.
And then people can look at it and go back. Yeah, it's very good work. Thank you. So I know Jesse missed out on Krishna's talk on the AI ethics or the positive feedback thing. So I don't know. Maybe Krishna, can you put your slides in the Slack as well to let Jesse take a look?
Jesse would have to see the whole meeting today because he was.
Oh yeah. Yeah. Well, you can go back and look at that too. But so yeah. Yeah, you missed it, Jesse. I'm going to go back and I'm going to do some things. If you have to leave at the top of the hour, go ahead. But I'm going to go through some things here. I'll go through our papers, but I also wanted to talk a little bit about this, a very short thing on this Euler cycles for life here. So let me go into this. So I presented this a long time ago, and I don't think, maybe people remember, maybe they don't. So this was a talk from, I think, last year or the year before that I gave in the meeting. And I'm not going to go through the whole talk. I just wanted to point out as a refresher what's going on here. So the idea is that Euler paths are similar to the Königsberg Bridge problem. So the Königsberg Bridge problem was a problem that was posed in about the 18th century in Germany, where there was this town Königsberg, and it had like 7 bridges that, because there was like an island and another island over here and some mainland. And they had a bunch of bridges, and the idea was, how do you cross each bridge only once, and still make the entire circuit? And this kind of is similar to the traveling salesman problem, where you have to visit a bunch of cities in the most efficient way. So that means only like going down one path once instead of doubling back. And so that's basically the idea here. An Euler cycle is a way to analyze these paths to make sure that you find the most efficient path. And so I put these seashells and diatoms in here because, as you'll see, this is like the kind of phenotype that we're kind of interested in, but also things like Volvox, which is a single cell colony, or it's a multi-cell colony of single cell organisms. So it's this it's another phenotype that might be relevant to this approach. And again, you see Euler paths in terms of like geometric shapes. So this is an octahedron, this is a dodecahedron. So you have a lot of like potential, see these partitions here, like kind of like cells, and then these blue lines are the paths between the cells. And so in a dodecahedron, you have a number of cells that maybe form a complex shape that has some depth to it, but you have these blue lines, and those are the networks. And so that's the idea. And you might ask, where?
Sorry.
Oh, go ahead.
Sorry. Are you going to look at bubbles, like a group of bubbles, to see where the vertices are?
Oh, I don't have a soap bubble. Yeah, I know what you mean, like the soap bubbles. Yeah. I don't have those in here, but that's a good point. I didn't bring that in. And there are some optimization problems having to do with soap bubbles, which if you ever looked at, like if you ever suds up a sink of water or a bathtub, you see like the suds form, and then these bubbles that form clusters, and they form these sort of hard edges inside that are not round; they're kind of square-like, and that's kind of a soap bubble cluster. And those actually are very similar to this thing as well. And I don't have the slides in here, but there's some optimization problems surrounding soap bubbles that if you look in the literature, you'll find. And so that's relevant as well. But why are cell boundaries important in living systems? And so we have these, all sorts of things happen at the interface between cells. We have these gap junctions that allow for electrical communication between cells. So we know, you might be familiar with synaptic connections between two cells, but those are chemical, but these are electrical connections, and they connect when two cells are near each other, and there's this connexin that allows things to cross the boundary between the two cells. It's a different form, and it's used in the nervous system. It's used in all different types of cells. So that's one reason why we're interested in these boundaries. Another is for, different types of nutrients, sort of a protovasculature system that goes in between the cells. In plants, you have this a movement of transcription factors and microRNAs that happens between plant cells, and so that's something that's important in plants. So there are a lot of reasons why we're interested in this. So the Euler circuit is an attempt to cross every independent edge of one of these networks. You can only cross each edge once, and that's the idea. And if you cross it twice, then it's no longer a unified network. It actually bifurcates into two networks, and you have to split it into two networks and analyze them. So the idea here is we want to find an Euler circuit, and that's how we define a unified system. And if we can't do that, in some cases, you can't solve certain networks. You can't find a complete Euler circuit. Then you have to divide it up into two networks. And so we can actually use a tool called Graph Online to calculate these. So we give it a network, and then we define the nodes, we define the edges, and then it evaluates the edges pretty quickly, and it tells you whether there's an Eulerian path or not. And so the idea would be that in evolution, you're adding these shapes. So you have triangles here. You start with one triangle, you add another triangle, you add another triangle, and eventually, you'll keep getting a complete Euler path. Or in some cases, when you add in another shape, and you keep replicating it, you end up with an incomplete Euler path. And in that case, then it divides into two different modules. And so then you start to have two different submodules that have to exist separately. That's the way those are the rules of defining this. So in this case, this network, which is actually a collection of different shapes, there are triangles here, but they're also squares. This does not give you a Eulerian path. And so depending on your generation rule for this, you can end up with these things that if we go back a few steps and take away some of these edges, we can have a Eulerian path. So this is how this works. You start off with a very simple shape. You start adding on, you maybe replicate a shape, or you add in different shapes, and you end up with something that has to be separated into two different modules, and then you keep growing a more and more complicated phenotype. So you end up with something like a seashell or a diatom, which is a very complicated shape. It has a lot of different elements in it. Some are triangular, some are circular, some are square, but they all have this sort of structure that we can evaluate like this. And so we can also look at things like rotation and scaling. And so in this case, we have a graph that forms a Eulerian path, but then we can distort it and actually form different phenotypes that are much more complex than the original. And then that also may change the transformation rules in terms of what gets replicated. So we can actually do this in a way that replicates more complex phenotypes. And so this is just a simple transformation rule here, where you're moving one node and you're stretching it out and you're stretching the entire network with it. And so the idea here is to evaluate very simple geometric shapes and geometric sort of the replication rules for different cells, and then stretch it out like you might see in morphogenesis to get these even more complex shapes. But we can still evaluate it using this simple methodology. And so that's all I'm going to talk about now. I was going to get into artificial genomes and hypercubes, but that's I think a bit much. I can go back to that later. So I wanted to just bring that up because I wanted to refresh people's memories on the origins of that work. And so it's still a work in progress. That's why I want to get out these submissions, because I want to improve that work somewhat. If you have any ideas on how to improve it, let me know. I've been kind of working on it, kind of in the background a bit, so I haven't spent a huge amount of time on it. And I started working on it in earnest about 2 years ago. So I think I'm going to go over maybe 2 or 3 papers here. The first one here is this paper in the Journal of Neurogenetics, and this is a perspective on C. elegans neurodevelopment, from early visionaries to a booming neuroscience research. And this is a paper that's, I think, more of a historical point. It just kind of goes into how C. elegans has been, how they started using C. elegans as a neurodevelopmental tool. So people will use C. elegans to look at nervous systems that are developing, looking at the different things that are expressed in different cells. We've talked about how C. elegans, we know every cell and its function, and in development, that's no less the case. We have a lineage tree where we know every developmental cell, and then what its ultimate fate is going to be. That's what they call deterministic evolution or deterministic development, where we know if we follow a cell lineage what that cell is going to become. Whereas in mammals, you get cell lineages where you get stem cells that proliferate, and then they respond to local signals to make to differentiate into functional cells. And so it's easy in C. elegans because not only do you have the deterministic system, but you also have the ability to trace through, and you have common, things in common. You have homologous neurotransmitters and things like that. So you can actually look at things like diseases, and you can look at those, aging, for example. You can look at all those things in C. elegans and get all the advantages of the C. elegans model organism, but also with the applicability to humans. And so it's, they talk about this in this paper. C. elegans has been mapped at the level of genes, cells, and synapses, providing the first metazone with a complete cell lineage, sequence, genome, and connectome. Here I summarize mechanisms underlying C. elegans neurodevelopment and the generation and diversification of neural components to their navigation and connectivity. I point out recent noteworthy findings in the fields of glia biology, sex dimorphism, and plasticity in neurodevelopment, highlighting how current research connects back to the pioneering studies by Brenner, Sulston, and colleagues. So Sidney Brenner was the first person to propose using C. elegans as a model organism. And before that, it was a very obscure organism. I mean, people wouldn't really think much of nematodes. Maybe if you're doing like soil science for understanding crop yields and things like that. But other than that, people weren't really interested in it. And Sidney Brenner was the first person to really say, we can use this as a model organism. John Sulston, of course, was the first one to map out the cell lineage tree of C. elegans. And so he sat there looking at a C. elegans under a microscope and actually drawing it out by hand, which seems, like a lot of work, and it was. But it gave us a lot of, it gave us a good basis for a lot of good science. So this review kind of goes through this. It gives you a timeline of the 20th and 21st century in terms of some milestones in C. elegans neurodevelopmental research. So if we zoom in here to this timeline, we'll see that there's in 1973 you get the first genetic screen and what they call defined mutants. People have investigated what they call the post-embryonic cell lineage, looking at some structural biology, looking at some anatomy. And then in the 80s, we got to the embryonic cell lineage, which was finalized in the early 80s. In 84, we had a physical genome map. And then in 93, people started using GFP, which is that green, fluorescent dye, or the green fluorescent signal that people use to look at different things in the cell. So you can express GFP with some gene and look at how it's expressed in a cell. So all these things came together like kind of gradually, and then all at once we had this great model for looking at different aspects of the biology. There was, people were starting to investigate the mechanisms of synaptogenesis in development, neuron rules for animal behavior. So C. elegans, you can also look at specific behaviors and really map it down to some specific neurons, which is not something you can do in humans, but C. elegans gives you that ability. And then, of course, going into the 21st century, you're getting whole genome sequences, like of the whole genome, so it makes it easier to understand what the defined mutants are all about, their location in the genome and everything, and then mapping it to humans because we want to know, for some of the disease research, how it maps back to humans in the human genome. So there's a lot of really cool stuff on this timeline. There's the male connectome a couple years ago, glia-borne neurons in 2014, nervous system maintenance back in around 2010, which is like, how do you keep the brain, how do you keep the nervous system from being overwhelmed by different proteins? If you're aware, Alzheimer's disease is caused by an aggregation of plaques and tangles of different proteins that accumulate in the brain, and so in the nervous system of C. elegans, you have similar issues with, maintaining the nervous system and the cells and their healthy state. So those things are really relevant to disease. They've done a lot of studies with microRNAs in terms of studying neuron fate. So they've kind of refined how neurons become sort of differentiated cells. It's they're deterministic, but there's also some variation in their function based on things like microRNAs and other types of neurotransmitters and things like that. So there's a lot of nuance in some of these mechanisms, even though they're so-called deterministic mechanisms. So this paper, it kind of really goes over a lot of this research. And if you go to the C. elegans meeting, if you attend that, there's a very strong appreciation for history in that meeting, where they talk about some of these names that you'll read about in this paper. So they talk about Solston and Horvitz, they talk about Brenner, they talk about some Marty Chalfie and some of these other people who really, did a lot of the sort of the groundwork for understanding C. elegans. Is this kind of, the sort of the complete understanding that we have, or somewhat complete understanding that we have of it today. So this is a nice graphic here that shows development in terms of cell birth and diversification, pathfinding of neurons when they send out their axons. They do pathfinding to find other cells to connect to, and then they have this synapse formation diagram, which shows a lot of the synapse formation that happens. So I wrote a paper about this, summarizing this as well, and this is a good, we have it down to specific times for these events. So a lot of the wiring of the chemical connectome happens after the egg hatches. So the egg, you get this morphogenesis within the egg, and then it hatches, and then you get this this finalization of the chemical connectome, or the synapse connections. Within the egg, there's actually a lot of this electrical connection between the gap junctions, but there's also a lot of this positioning of cells and migration of axons and things like that. So just so you have a timeline for that. And so this kind of goes through the nervous system, formation, and the history. So this is a very good paper if you're interested in that. I would definitely read it. I'll go through this paper. What else do we have in here? We have this paper called Physical Constraints on Early Blastomere Packings, which is another paper on it's something that I don't know who sent, maybe I found this paper, I don't remember. But there's an abstract. Let's read the abstract on this. This is a C. elegans relevant paper as well. So this is about something called Blastomere Packings. And the abstract reads that at very early embryonic stages, when embryos are composed of just a few cells, establishing the correct packing arrangements or contacts between cells is essential for the proper development of the organism. As early as the 4-cell stage, the observed cellular packings in different species are distinct, and in many cases differ from the equilibrium packings expected for simple adherent and deformable particles. So this is about the physics of the early embryo and how the cells are arranged, and then explaining contacts between those cells in that way. So and this is not just a random thing. This is very specific to the function of the cell and the later parts of embryogenesis. So it is unclear what are the specific roles of different physical parameters, such as the forces between blastomeres, their division times, orientation of cell division, and embryonic confinement, play in the control of these packing configurations. Here we simulate the non-equilibrium dynamics of cells in early embryos and systematically study how these different parameters affect embryonic packings at the 4-cell stage. In the absence of embryo confinement, we find that the cellular packings are not robust, with multiple packing configurations simultaneously possible and very sensitive to parameter changes. Our results indicate that the geometry of the embryo confinement determines the packing configurations at this stage. Removing degeneracy in the possible packing configuration and overriding division rules in most cases. So overall, these results indicate that physical confinement of the embryo is essential to robustly specifying proper cellular arrangements at very early developmental stages. So this is something that I think Susan would be interested in following up on. Maybe she is interested in a lot of the stuff related to embryo physics and like the sort of the packings of cells, as I recall. So this paper kind of gets you down to the very early stages of embryogenesis, something they call the blastomere. So it's really when you have just these founder cells of the different lineages packed together. And so you can study this in C. elegans. So in the nematode C. elegans, they sort of form a diamond shape. In mouse embryos, they form a tetrahedron. And in sea urchins, cells arrange in a square configuration. And so depending on the organism you go to, even at that stage, they form a certain geometry. And it's not necessarily the same one. You were going to say something?
Okay. Yes, I've just been looking at human development, and they actually develop as single rounded cells for the first 8 divisions or so, and they go one cell at a time. So instead of going from 1 to 2 and from 2 to 4, they actually go from 1 to 2 to 3 to 4, and they're all round. So it's interesting.
Yeah, it's definitely. And then I guess the implication here is that you have this. It sets up the rest of the pattern that you're going to find in the. I mean, I think that the assumption is that it's just that there isn't much to do with shape that early on in embryogenesis, but I guess they're finding that that's actually not really true. That there's a lot of variation. But they do a lot of I think they do a lot of simulation in this paper. They kind of consider the contact angle and all these other things that are, and they look at mouse C. elegans and C. urchin. So they're doing this wide range of phylogeny here. So you can see that what they mean by the angles here. So the C. elegans, you have this diamond. So you have 2 cells here, top and bottom. You have one that's anterior and one that's posterior. And then in mouse, you get this triangle shape. And then, sea urchin, it's squared off. So you actually, in sea squirts, I know, from some of our earlier work, they sort of butt into 2 cells. They have this sort of 2-cell, they just sort of replicate and then stay in place. They don't really move like anterior, posterior, like C. elegans. They just have this sort of square packing shape like this. It's very square. And then they also have a lot of asymmetric divisions. So like in the human case you mentioned. And then they form, later on they're going to form this pretty complex morphology. But in this stage you're just doing it in squares. They're just going in one orientation at a time. So it's really interesting.
An interesting stage in vertebrate embryos is the part where they pack together, or I guess it's mammalian embryos form a morula, and that's when cells pack together. They kind of cramp themselves together. Before they're more loosely associated.
Yeah.
And then they form a morula.
Yeah.
Anyway, yeah.
That sounds good. Yeah, I mean, this is something to follow up on, I guess. I mean, we've done some work on this, but we've not thought about in this way. So they actually do some numerical simulations in here, and they look at some of the aspects of cell-cell interaction potential. So they're looking at different parameters, the confining shell. So they're doing a lot of mathematical modeling in here, and then so this is. And then they're doing this sort of simulation here where they're generating these different shapes and over time, and they're looking at how they order. So yeah, this is a really good paper. it's actually an uncorrected proof. So there might be the actual corrected proof might be out by now, because I think I found this paper a couple weeks ago and put it in the list. So this is and I'll provide a link to this later on this folder. The last paper I'd like to talk about today is what is this one? I don't want to do that one. I'll talk about I think Krishna had to leave, but there's a paper on hub connectivity, neuronal diversity, and gene expression in the C. elegans connectome. And that might be something interesting for the difference between artificial neural networks and biological neural networks, because artificial neural networks are these representations that just have these cells and they're connected, but we're kind of approximating just the electrical or the chemical activity between the cells. And so, that's the only thing we're really interested in. But of course, nervous systems are much more detailed than that. They have a lot of things going on. They have these different motifs in their connectomes, and one of those is this hub connectivity, which is where you have certain neurons that connect to many other neurons and form functional hubs. And so you have this. They've looked at different brains, not just C. elegans, but mouse and humans. And so you can find these hubs in any kind of nervous system you want. They call them rich club neurons because they're richly connected relative to other neurons. And so, again, these are these chemical connections. And so, in this case, they look at C. elegans connectome that look at like how gene expression is coupled to these differences in connectivity. And so, they looked at 279 C. elegans neurons and binary gene expression data for each neuron across 948 genes. So in C. elegans, that's a fair amount of the genome. We computed a correlated gene expression score for each pair of neurons. So they're looking at the connections between neurons and that expression score, and then they're looking at different things in terms of gene expression with signaling and different types of signaling between neurons. So glutamatergic and cholinergic signaling are independent processes that we might be able to pull out of these data. And so we further show that coupled expression between hub neurons cannot be explained by their neural subtype. So just because two cells are connected and they're expressing the same things, it doesn't mean that they're of the same neural subtype. It just means that there's something that they're doing maybe autonomously as cells or independently that are, defining this sort of common expression pattern. It's not just because they're of the same cell type. Also separation distance, chemically secreted neurotransmitter birth time, pairwise lineage distance, or topological module affiliation. None of those things also explain the data. So their results suggest that neural hubs may possess a distinctive transcriptional signature themselves, preserved across scales and species that are related to the involvement of hubs in higher order behaviors. In other words, these hubs are sort of evolutionarily conserved, and they serve a function in the network, which is to organize a lot of the connections, and they express their own gene expression patterns, and maybe because they're hubs, they do this. And so it's a kind of an interesting paper. It's definitely something we don't even think about in neural networks, but it might be something important to understanding like why there's this topological diversity. And so this paper goes through, it's very biologically oriented, but it kind of goes through the logic here, combining the data sets and then giving a result here. So they have they show the hub neurons constrained within the head and tail. So they show the position of the hub neurons versus the other neurons. Then they show hub connectivity, and then they show this. So they show that this exists. This is a well-known result, but you also get this rich club organization, and then you get these correlated gene expression values. So now they're looking at gene expression in these cells, and they're looking at rich club cells versus feed-out cells versus feed-in cells versus peripheral cells. So I don't really know what those categories are, but they're in the paper. And they kind of have these classifications of cells here. You have these different classes of cells, and you see there's a rich-to-rich connection. You have other, like, peripheral cells to rich connections, and those are all kind of summarized in this table. So the idea is that the rich club is unique in many ways, and that there's maybe some functional reason for that. And so, so it's a very good paper. Again, it's very biologically oriented. And again, here's the link to the drive. So if you're interested in following up on any of these papers, you can. Let me see what we have in the chat here. So we have, these are the slides for the ethics abstract from Krishna. This is for anyone who wants to look at the slides here. We also have, Jesse says, thank you. Krishna had to leave, that's fine. And then here are the links. Here's the link to this folder of the papers. So any questions before we go on anything today?
I have a minor comment.
Okay.
I think I'm going to try to do a similar thing from oral and make a little doTERRA folder from pilot papers of DevoWorm, people that do an idea. Because having a reference is helpful.
Yeah, we've been doing this with we've already been doing this a little bit with EndNote, and so we might do it with EndNote. We might do it with doTERRA. I'm not really sure yet, but I think we already have one with EndNote. We can talk about it later, though.
Yeah, let me know, because I know that I was talking about it, but then I sort of seen it. But that's good.
Yeah. Well, we haven't been keeping it up, so I mean, we have to go back and maintain it. So.
Okay, but no, it was really good. I'm going to go back and look at Shruti's presentation, because that was really interesting and related to things that, like, I kind of did some things related to, like, effective computing before and getting the tone. It like remind me of that. So I would check that out. And I did a quick glance at the AI ethics slides for Krishna, and they look good. There was actually some really interesting overlap between one of the things that I found on the podcast this week, and mentioned very briefly in terms of his feedback component. Like there's some things to follow there. So it was really good, and I'm going to go back and look at the whole meeting. So thanks for all of that.
Okay, well, thanks. Any other comments? Oh, we got one in two in the chat. Okay. Okay, yeah, thank you from Jesse, and Shruti has her link here to her. Okay. So, yeah. Okay. And I'll share those links, too, if people are in need of them. So, okay, yeah, thank you, Mainak, for attending. Thank you, everyone, for attending. And we'll be back next week. We'll talk more about some of these topics. If you want to present, again, you can bring it to the meeting. We'll put it on the list, and we'll go through it. And there's so many different topics here. And we'll also keep on top of the deadlines. So no immediate deadlines except for that Waves special issue, and we know about that. So, all right.
Okay, well, thank you, and I'll send you a picture of bubbles.
Okay, thank you. Yeah. All right, everyone. Have a good week.
Bye, everyone.
Bye.
Bye.